Hello, welcome to the BSI Regional and Affinity webinar, the next in the series. Today it's run by the BSI Tumour Immunology Group together with the National Cancer Research Institute. So I'm Benny Chain, I'm chairing the session, and I'm a Professor of Computational Immunology at UCL. Let me start off by thanking Miltini Biotech, who are the sponsors and principal supporter of the BSI Regional and Affinity Group program events. Thank you very much. These are very valuable part of the immunology calendar. So today the webinar is entitled "What Do Cancer T Cells See?" And we'll have two speakers, Professor Lindy Durrant, who's Professor of Cancer Immunotherapy from Nottingham, who's going to be telling us about peptide modifications as tumor antigens, and Dr. Michele Mishto, who's the senior lecturer of immunology at King's, and who's going to be telling us about peptide splicing. So we'll have two talks of 20 minutes. I'll try and keep an eye on the time so we don't overrun, and then leave 15 minutes for questions and answers at the end. So attendees are on mute because there are too many people to have questions. So if you could send the questions using the questions function, I'll try and ask the questions on your behalf at that point. So just before we hand over to the speakers, I'd just like to introduce you very briefly to Robin Taylor, who's the journal manager for the BSI, and he's going to tell you about a new open access journal that the BSI are launching. So Robin, over to you.
Thank you, Benny. Yes, I just wanted to jump in here and tell you a bit more about our new launch journal from the BSI, Immunotherapy Advances. It's our first open access journal that we have launched as a society, and it launched in December last year, and we already have some really excellent articles if you want to check them out. It's edited by Tim Elliott of Oxford University, and we are supported by a wonderful editorial board. The journal focuses on the translational pipeline for immunotherapy from discovery all the way to clinical trials. We're interested in any kinds of articles, particularly first in human clinical studies and negative trials as well. I just wanted to introduce you to some of our regional editors. We've built a wonderful editorial board focused on trying to be as international as possible. So we focused on having someone that represents from each each continent, as well as different subject areas within immunotherapy, as well as our editor-in-chief. We have over 40 members of our editorial board as well, from all areas of immunotherapy and from all over the world. If you're interested in submitting, these are our main article types, and something we're really keen to promote in particular is our Trials Watch series, which focuses on summarizing and discussing some recent clinical trials within immunotherapy. We've recently published one written by Gary Middleton in collaboration with the Tumour Immunology Group, which focuses on recent clinical trials in melanoma. But thank you very much for your time, and we really appreciate if you would be more interested in, if you'd be interested in hearing more about the journal, please check out our website or feel free to get in touch with us. Thank you.
Great. So we'll, without any further delay, thank you very much, Robin, and it sounds like an exciting new venture. I will hand over to Professor Lindy Durrant for the first talk. So if you could, 20 minutes, try and keep to the time, that would be great. Thank you.
So thank you very much to the organisers for inviting me to give the lecture today. What I'm going to talk to you about is stress-induced post-translational modifications as targets for cancer vaccines. So what I'm going to talk to you about predominantly today is the post-translational modification citronellation. And citronellation occurs basically in a polypeptide when arginine is converted to citrulline. And that happens by a series of enzymes called PAD enzymes. And in order to activate the PAD enzymes, you need high levels of calcium. That can clearly be during apoptosis, when the membrane becomes permeable and you get the calcium rushing in. But for the purposes of the talk today, we need activated PADs in viable cells. So in viable cells, that can only occur in double membrane vesicles. So that can occur in the nucleus, and indeed citronellation is an epigenetic mechanism for controlling genes. But for our purposes today, the vesicle we're interested in is the autophagosome. So the autophagosome or autophagy is induced in stress cells as a way of degrading some of the cytoplasmic antigens in order to generate new energy. Within those autophagosomes, you can get high levels of calcium, and you can gain this conversion of arginine to citrulline. When that happens, a lot of the serum proteases within the autophagosome cleave after an arginine. So if you change your arginine to a citronellline, you will no longer get cleavage at that site, and you get what we call a novel peptide being expressed. Now, the class 2 processing process all the time samples the autophagosome and looks for epitopes it can present. So clearly during stress, when you've got these activation of PADs, you're presenting a new series of peptides, but those peptides also contain a citrulline. Now, as you're probably aware, most cells don't express MHC class 2, but indeed most of them can be induced to express class 2 with gamma interferon. So in order for this process to work, we need two things. We need the stress to induce the autophagy, but we also need a degree of inflammation to upregulate the MHC class 2. Now, this process has been well known and for some time in rheumatoid arthritis, but what we were intrigued to find out is we know most tumour cells are stressed and have high levels of autophagy. So the question really was, are they presenting citrullated epitopes, and could they be good targets for a cancer vaccine? So what we did is we made a series of citronellated peptides. We used them to immunize HLA transgenic mice. They stimulated CD4 T cells. Those CD4 T cells went into the tumor environment where they initially recognized antigen-presenting cells. Those antigen-presenting cells had taken some of these citronellated proteins from some sort of necrotic tumor. That basically activates the CD4 to produce gamma. The gamma then can upregulate the MHC class 2 on the tumor. And as I say, the tumor all the time is making these citronellated peptides. So they now get presented on the surface of the tumor and become targets for these CD4 cytotoxic T cells. So that's the proposed mechanism of action. Let me try and convince you with some of our data that that is indeed what's happening. So the vaccine I'm going to talk to you about, we call MODI-1. It's a combination of two citronellated vimentin epitopes and an enolase epitope. Again, these targets are modified, they're citronellated, but these proteins are very good fuel for autophagy because they're very ubiquitous cytoskeletal proteins. So this is, again, it's in the HLA-DR4 transgenic mice, and you can see if we immunize with the citronellated peptide, this is one of the vimentin epitopes here, we get a lovely ELISpot. This is a gamma ELISpot response, but it doesn't cross-react with the wild type. Now, there is indeed one amino acid difference in this peptide between human and mouse. So we've also used the self, the mouse epitope. Again, you can see a nice response to the citronellated epitope, nothing to the wild type. The second peptide here is completely homologous. So we see beautiful responses to the citronellated and not to the wild type. And again, with enolase, we see nice responses to the citronellated and not to the wild type. Okay. So definitely there is a repertoire in these HLA-DR4 mice to these citronellated epitopes. So we went on then to see in the similar model, could we see anti-tumor responses? So in this B16DR4 HLA transgenic, we give the tumor on day 1, and then 4 days later, we immunize either with the citronellated or the wild type peptides. So you can see here when we immunize with the human citronellated peptides, we're seeing extremely good survival in these mice. They completely reject all the tumor and the majority of the animals survive. In contrast, if you use the equivalent arginine wild-type peptides, there is no significant anti-tumor response. Again, as I said, because a couple of these peptides had an amino acid difference between human and mouse, we used the self-peptides, the same urine peptides. Again, you're seeing all three citrinated peptides give significant anti-tumor responses, whereas the wild type give no response. So this would suggest indeed tumors are stressed, are undergoing autophagy, and do indeed present these epitopes, which could be good targets for a cancer vaccine. But clearly it would be quite nice if we could cure everybody with DR4 from getting tumors, but it would be even better if we could look at other HLA types. And indeed, if this is a normal stress response, you would expect it to work in other HLA types rather than just DR4, or there would definitely be a selective advantage in being DR4. We've used our DP4 transgenic mice, mainly because DP4 is quite a common HLA antigen expressed by quite a number of individuals. And indeed, we do see nice responses in DP4, but we have no reason to believe it's just restricted to DP4. Again, just showing you here, nice responses to the citrinated epitope, not to the wild type. Again, very nice to the citrinated, not wild type, for all three epitopes. So we do think these peptides can be presented by multiple HLA types. We then again went on to look at the anti-tumor response in the DP4 mice with a DP4 expressing tumor as well, which is what we're looking at here. And we've also added a further level of complexity here, because we've put the class 2 under an inducible gamma-inducible promoter. So this is what you would expect to see in the majority of tumor cells that don't normally express class 2, but can be induced by gamma interferon. And you can see in these inducible models, either in the DR4 or the DP4, we're still getting superb anti-tumor responses with this combination of these three citrinated epitopes. We tried to work out what cells are actually involved in the anti-tumor response. Is it the CD4s that are setting up a pro-inflammatory environment, but it's the CD8s that are effector cells, or indeed are the CD4s directly recognizing the tumor? So again, this is the IDP4 model. We give the tumor, we immunize, and you can see we see a nice anti-tumor response. If we deplete the CD4 cells in this model, you can see we completely lose the anti-tumor response. So indeed, the CD4 cells are vital to this anti-tumor immunity. Probably much to our surprise, though, when we depleted the CD8, it made little impact on the anti-tumor survival, suggesting that in this particular model with these epitopes, CD8s have very low, if any, role to play in this anti-tumor immunity. We went on to see, is it actually dependent on citrinylation and autophagy? So again, here we're immunizing DR4 transgenic mice, and then we're looking at ELISpot responses against a variety of different targets. What you can see here is if the tumor lines do not express DR4, so they're mismatched at class 2, you see no recognition whether the cells are happy or stressed. If you look at the appropriate HLA matched target, so the DR4 target, healthy cells are not recognized, but stressed cells are recognized extremely well. This is also true for lymphoma cells, happy no recognition, stressed recognition, pancreatic, happy no recognition, stressed recognition, or this is indeed a Lewis lung carcinoma line, happy no recognition, stressed good recognition. Again, if we take this one stage further and we look at recognition again of this serum-starved DR4 line, and then we incubate in the presence of either an inhibitor of autophagy or a PAD inhibitor or both, you can see they all significantly reduce the immune responses. Indeed, also, if we look in where we induce autophagy with rapamycin, we see lovely recognition. Whereas if we do it with valfamycin, which does induce autophagy but blocks the pathway to class 2 presentation, you can no longer see that recognition. So these results would suggest that indeed the anti-tumor immunity is dependent on citrinylation in the tumor and autophagy within the tumor. We also went on to see do the tumor cells themselves have to express MHC class 2, or indeed were the CD4 T cells being stimulated in the tumor environment, but then something like NK cells or something was the main mechanism of action. But hopefully you can see here, if we have B16 that do not express MHC, cannot express MHC class 2, they still have the appropriate MHC class 1. but not MHC class 2. And again, 4 days later, we immunize. Now we have completely lost the anti-tumor response. So although the APCs within this model can still express MHC class 2, because the tumor no longer expresses it, we do not see an anti-tumor response. So this would suggest that the CD4 T cells are looking at direct recognition of MHC2 in these tumors in order to have an anti-tumor effect. We went on to see whether these targets would be appropriate in human tumors. So we've looked for vermentin enolase. We have actually an antibody that recognizes citrinated vermentin and citrinated enolase. And you can see here a nice strong expression in a whole range of tumors. This is just giving you an example. We also looked for MHC class 2 expression, and you can see quite a number of tumors actually do express MHC class 2, and this is without the vaccination, so this is just in the normal tumor growth. They all express pretty much PAD2 and PAD4, and most of them have high levels of autophagy as recognized by AMBRA or P62 staining. So when you're looking at these various tumors, you can see that breast cancer, renal cancer, ovarian cancer, and head and neck cancer potentially all have high levels of the markers that are important in being able to express these citrinated epitopes. We also went on to see, is there a repertoire in humans that can recognize these citrinated peptides? So this is just naive human donors or blood from cancer patients where we incubate them with the antibody. The cells are CFSE labeled, so clearly if they proliferate, they lose CFSE, and you see this population down here. And it's just showing you some data from 2 ovarian patients that you can see respond to all 3 of the epitopes, giving nice CD4 proliferative responses. Indeed, if we look across a range of patients to the 3 epitopes, both normal donors and cancer patients respond quite nicely to these epitopes, and there's no significant difference between the 2. So there's definitely a repertoire in humans. We did some initial phenotyping work, and you can see that they express gamma interferon. They also express granzyme B, suggesting they have cytotoxic potential, and they also express the activation marker CD134. Because we were seeing such good anti-tumor responses, we went on to see whether we could push this survival a little bit better. So in essence, what we were doing is giving the tumor at day 1 and immunizing at day 4. We extended that period, so we didn't immunize until day 7, day 10, or day 14. And what you can see here is this is the day 4 response to the epitopes. Nice anti-tumor. At day 7 and 10, if anything, it's better, but it's certainly still highly significant. And even if we wait till day 14, and that's when some of the animals in the control group have to be culled because their tumors are so large, we are still seeing an anti-tumor response. So this is a fairly powerful immune response that works against even very bulky tumors. And that's true whether we do the HLA-DR4 model or the HLA-DP4 model here. Again, we're still seeing good anti-tumor responses at day 7 and day 14. So we then took this one stage further, and that what we actually did is we waited till tumors reached about 5 by 5 or 8 by 8 in size. So we definitely had tumors. And then we immunized. We didn't immunize the tumor, they're immunized basically systemically. But what you can see here is if we immunize with the black dots, the majority of tumors then do regress if they're immunized with MODY1. And that's shown here on this waterfall plot, where you can see the majority of tumors immunized with MODY1 do regress. In contrast, this is just the adjuvant control, just using CpG and MPLA. And you can see when we immunize the animals with these, the tumors continue to grow, again, as shown here by the waterfall plot. What we tried to do here is when these tumors were regressing, we took them before they disappeared completely and then tried to analyze their T-cell infiltrate. And what you can see here is, again, in the MODY1 group, there's nice high levels of CD44, nice high levels of CD4, and good levels of CD8 in comparison to the adjuvant alone control group. If you analyze that in terms of specific cells, what you can see interestingly, as the tumors regress, we get more and more CD4 cells, suggesting they are the main effector cell. In contrast, interestingly, the number of CD8s as the tumors regressing decrease. And interestingly also, the myeloid-derived suppressor cells, or CD11b GR1 positive cells, also go down as the tumour regresses, suggesting possibly we are flipping that immune environment by sending in these CD4 T cells. To take this forward into the clinic, rather than using mixtures of peptides and adjuvant, we've actually now linked the adjuvant to the peptide. And we've been using this adjuvant Amplavant from Case Malief's group. It's basically a TLR1/2 adjuvant. As I say, we link it directly to the peptide. Hopefully you can see here, as you reduce the dose, we still see lovely responses with the linked peptide, but the responses to the mixture drops off very quickly. And indeed, we have between 100 and 1,000-fold better immune responses by linking the epitopes or the peptides to the adjuvant than if we do in the mixtures. And this is really good news for the clinical trial, because now it means that we can go with microgram amounts of antibody into patients rather than milligrams, which have the potential to energize T cells. Again, that's just showing you some of the data. This is either linking with a TLR9 adjuvant or the 1/2 I talked to you about before, where we're still seeing good, strong anti-tumor immunity. Again, if we take these animals that have initially rejected the tumor and re-challenge them with tumor with no further immunization, you can see they have a nice memory response, and they do reject that re-challenge. So we do seem to be inducing good T cell responses, good CD4 responses, and they are memory responses.
About 3 minutes left, Lindy.
Yep, nearly done. Yeah, so we've shown in the past that we've seen no difference. We get good anti-tumor responses. We don't really need checkpoint, but who knows in human cancer. So for this, basically, we've used the day 14 model, see a nice response to the vaccine, nothing to the PD-1, but nice synergy between the two. And that's the clinical trial that we're now going to go forward with. We're going to try a couple of doses to optimize the immune responses. We'll go into these cancers that we've identified, either as a monotherapy or for head and neck, potentially in a combination with a checkpoint inhibitor. So just to summarize then, the responses are citrullinated specific and do not recognize wild type. It's CD4. You do need MHC2. It's dependent on gamma interferon. At the moment, we see no role for CD8s. They do need autophagy, and they are widely expressed cytoplasmic antigens that are modified or citrullinated. That's by the PAD enzymes. Seems to be cytotoxic CD4s. It works in very bulky tumors, and it generates memory responses, and there is a repertoire in humans. So finally, just to thank the team that did all this hard work for me, and thank you for your attention. I think we'll now press on to Michele, who will do his presentation.
Can you hear me?
Yes.
Yeah.
Good afternoon, everybody. I would like to thank the organizer of this webinar for inviting me. Today, I'm going to speak about unconventional epitopes and how they are, which unconventional epitopes are recognized by cancer T cells. Unconventional epitopes are kind of a hot topic in immunology in the last decades, even if they've been described for the first time much earlier. I define as unconventional epitopes in the context of the MHC class 1 antigen processing and presentation pathway, all those epitopes that induce an immune response and that they do not derive from linear sequences of known proteins. Among them, for example, we have cryptic peptides, so that are peptides that derive from cryptic non-coding regions, for example, endogenous retroviral virus sequence or 5' UTR. Other unconventional epitopes are those epitopes that carry a mutation, and this is usually very important for somatic mutation that occur in cancer. It's called neoepitopes. That are also modified with post-translational modification that has been already described in part by my esteemed colleague. This modification can occur both at antigen level, at peptide level. And then there are the post-translationally spliced epitopes that are the topic of my talk today. Indeed, proteasomes, that is the one of the main proteases in eukaryotic cells. When you switch off the proteasome, the eukaryotic cells usually dies. So the proteasome is degrading the antigen, releasing the peptides in the cytosols. Some of them survive the aminopeptidase destruction. They are translocated into the ER, mainly through TAP. They bind the peptide loading complex, and if they have the right sequence to fit into the cleft of the MHC class 1 molecule, then they form a stable complex that is translocated to the cell surface, where they can be recognized by the CD8 T cells. So the proteasome is able to ligate to non-contiguous fragment of an antigen, and this activity is called proteasome catalyzed peptide splicing. If the two fragments derive from the same antigen, we define this process as cis peptide splicing, that can occur in a normal or reverse direction based on the fragment orientation. And if the two fragments derive from two different antigens, we call this process trans-peptide splicing. The research in the field has been kind of limited by the fact that spliced peptides, since they are not present as such in the proteome, I have some difficulties in their identification. Therefore, all groups that they wanted to study them had to develop their own method for the identification and the characterization. The research started in 2004 when two different groups, both stimulated by the idea of Jonathan Yodel, identified the first two spliced epitopes that they were recognized by tumor infiltrating lymphocyte. Since then, we had various achievements in different aspects of peptide splicing and in terms of translational application in cancer, infection, and autoimmunity. I will today analyze few of the results that we and others obtained in the last years. First information that I would like you to keep in mind is that peptide splicing is a frequent reaction. So last year, we published what is at the moment is the largest database of products generated by proteasome in vitro digestion. We have synthetic polypeptides derived from antigens that are digested by purified proteasomes, and then the products are detected and in some cases quantified by mass spectrometry. And in a pool of over 50 substrates, we identified a number of spliced peptide that is on average superior to the number of the non-spliced peptide. So the peptide splicing is a frequent mechanism, it's not like an exotic activity of the proteasome, and also is not a random process. We developed a method for the quantification of spliced and non-spliced peptide in this in vitro reactions that allowed us to quantify the frequency of cleavage of proteasome here in orange and the frequency of splicing of the proteasome in blue in a given substrate. Here I just show you KRAS polypeptides. And as you can see here, peptide splicing does not occur everywhere, and also there isn't a direct correlation between the usage of a given site for cleavage and splicing. And this suggests, therefore, that the two reactions, they are in a kind of competition among each other. And indeed, there's the same catalytic site of the proteasome that catalyze both reactions. And based on the sequence, this is our hypothesis, then one product is released by peptide hydrolysis, and another one is released by peptide splicing. We also developed method for the identification of spliced peptide in the immunopeptidomes, so the peptides that are bound to the HLA class 1 molecules. We identified many spliced peptides. For example, this is a study that we published in 2019 analyzing the HLA class 1 immunopeptidome of cancer cell lines. And what it was interesting, it was to see that not only peptide splicing increased the variety of the peptides that are in the immunopeptidome, but also enlarged the antigenic landscape. around 20% of the antigens that we detected in the immunopeptidome, they were represented only by spliced peptides. And these antigens, they have some characteristics because they were longer, more hydrophobic, and more basic than the antigen that they were represented only by canonical non-spliced peptides, suggesting that this characteristic helps or promote the catalytic splicing reaction. If we move on the immunological relevance of the spliced peptide, I told you that there are already evidence since 2004 that tumor-associated spliced epitope are recognized by tumor-infiltrating lymphocyte. So we perform the first study on studying infection. We had a mouse model of Listeria monocytogenes, that is a bacterium that is released in the cytosol of cells, so intracellular bacterium, that triggers CD8 T cell response. We developed methods in silico that led to a final list of spliced epitope candidates that derived from PLCA and PLCB, that are two antigens of Listeria. These antigens, in at least they are predicted to be not really good to don't lead a clear immunogenic response by CD8 T cell in the black 6 mice, because there are not good canonical non-spliced peptides that bind the 2KB. So what we did, we had a list of the epitope, we infected the mice, we took the spleen, isolated the splenocyte, and co-cultured them with the epitope candidate that we had from the in silico study. And then we measure the CD8 T cell response by FACS analysis of interferon gamma. As you can see here, we have LLO 296-304, that is our positive control, is a known non-spliced epitope that is derived from another antigen of Listeria. And indeed, in infected mice, we have a response. And we had a response also against two spliced epitopes derived from PLCB, out of the around 20 spliced epitope candidates that we test. The response is specific because we don't have a response when the mice are not infected. And also the splenocyte of infected mice do not recognize the spliced peptides that share part of the sequence that is present in the spliced peptides. So the response is specific towards the spliced peptide. And this is even more clear in the experiment that we performed during the revision, where we co-cultured the splenocyte of infected mice with EL4 cell that they were transfected with LLO antigen, and then we measure a response as our positive control. We have a response also against the PLCB, but when we mutated the anchor site of the spliced epitope in PLCB, and this is the PLCB delta, we didn't have a response anymore, therefore suggesting that these two spliced epitopes in this mouse model drive the immunogenic response towards the PLCB antigen. If we move on tumor, we described for the first time in 2016 a natural response in melanoma patients against two tumor-associated derived from spliced epitopes derived from GP100. As you can see here, the response was detectable in half of the patients. When the patients were treated with a vaccine of an antigen unrelated to the GP100, then we had less positive patients. And we also have one response in a healthy control. The analysis was done on interferon gamma ELISA and confirmed by FACS analysis. And recently, last year, a paper of Purcell Lab confirmed our results that indeed there is a response in melanoma patients, natural response. But I think that for tumor immunology, a very interesting study was what was published by Rosenberg Lab in 1994. They treated a melanoma patient with autologous tumor-infiltrating lymphocyte, one clone, that recognized tyrosinase, that is a melanoma-associated antigen. The two treatment of adoptive T-cell therapy led to the complete regression of the tumor. At the time, they didn't identify the epitope recognized by the clone, and almost 20 years later, Benoit van den Eijden and colleagues proved that that clone recognizes a spliced epitope and does not recognize any other non-spliced peptide present in the tyrosine. So therefore suggesting that spliced epitopes could be used in adoptive T-cell therapy in cancer. Indeed, then following this hypothesis, we develop a method for the identification of antigen and mutation in the antigen that could be interesting for the identification of spliced neoepitope. We then had, applying this method, we identified KRAS and the KRAS G12V mutation as interesting, and then we verified the production of the spliced epitope candidate in vitro assays. We performed an in vitro digestion of the proteasome of the KRAS antigen, and we identified by mass spectrometry the KRAS G12V spliced epitope reported here. As you can see here, the in vitro digestion and the synthetic peptide are very similar, so confirming our identification. But we also investigate other aspects of the antigen presentation pathway. For example, the top affinity with the peptide, that was very good, so that was suggesting that this spliced epitope could be translocated in the ER by TAP. But also the binding affinity to HLA-0201, that is the most frequent allele in Caucasian population. And as you can see here, both the predicted and then confirmed in collaboration with SETE Lab binding affinity of the spliced epitope was quite good, around 70 nanomolar. In collaboration with Blankenstein Lab at the Max Delbrück Center in Berlin, then mice with the HLA-0201 and TCR alpha beta have been immunized with the epitope. CD8 T cell has been isolated, cloned the TCRs, transfected in CD8 T cells that they were then co-cultured with cancer cell line that are HLA-0201 positive and either express the wild type KRAS or the mutated KRAS. As you can see, only the mutated KRAS present in the SW480 cancer cell line led to a specific response of the CD8 T cells, therefore confirming that proteasome produce spliced neoepitope. It produces it in an amount that is sufficient to be presented at the cell surface by cancer cell line and trigger a response by the CD8 T cells. Therefore suggesting that TCRs specific for this spliced neoepitope could be useful for adoptive T-cell therapy of a large cohort of patients because this mutation is very frequent in pancreatic adenocarcinoma and colorectal cancer. The problem, so one of the issues that trouble the scientific community, however, is the real theoretically very large sequence variability of spliced peptides that can kind of clash with our model of central and peripheral tolerance and overlap between self and non-self. Because if we have like a very large number of peptides that are spliced peptides presented at the cell surface, this could lead to the elimination of CD8 T cell clones and therefore a reduction of the repertoire of CD8 T cells in periphery that, for example, could limit the response towards viral infection. So we started last year to analyze in silico to quantify this risk. We developed a large database of spliced peptide derived from the human proteome as well as derived from around 100 viruses, and then using different type of filter we try to understand which was the amount of Twitter peptides. So Twitter peptides are peptides that they have exactly the same sequence, either if derived from self or if derived from a virus. Okay, so they are virtually not it's virtually not possible to distinguish them, their origin, by CD8 T cells. So if we consider all the potential combination of splice and non-splice peptide, the amount of zwitter, so the frequency of zwitter peptide, is quite limited if we consider only non-splice peptide. But on the contrary, around 4% of all the theoretical sequences derived from the splice peptide could be zwitter peptide, as reported here. When then we consider also the binding affinity to the HLA-0201, that is the most frequent allele in Caucasian population, as well as the expression of the antigens in the MTECs, and so the MTECs are those that present the epitopes to the thymocyte during the negative selection, then we still have like a very few cases of non-splice peptide, but like a significant number of spliced peptide that are zwitter, or it could be zwitter peptide. However, what we know from both our in vitro experiment and the immunopeptidome analysis is that only a tiny fraction of the all potential splice peptide sequences is then generated by proteasome. So when we included this information in our in silico analysis, and then we computed the number of, the average number of zwitter peptide per virus, then we had some example for non-splice peptide, but only when we hypothesized a very large frequency of splice peptide in the immunopeptidome, then we can detect, or we can estimate a relevance of splice peptide in the generation of zwitter peptide. This first preliminary, very preliminary study that does not consider many aspects of the antigen-protein presentation pathway, therefore suggests that splice peptide may not impinge upon the T cell repertoire. However, it's always possible.
About a couple of minutes left, Michele, if you could come to the end.
However, it's always possible that a T cell, potentially autoreactive T cell clone, survives the negative selection, and that this could lead to an autoimmune response if this epitope recognized, if this clone recognized, for example, zwitter peptide that's derived from virus or human. And in silico study that we are conducting this period, we identified examples that are related to type 1 associated, to type 1 disease. For example, epitopes that derive from a tumor-associated type 1 associated antigen, as well as a type 1 associated virus. All this has to be proved that is true. It's just in silico study. And then I finish my talk thanking all the people that have been involved. Among all the collaborators, it's very important to thank Juliane Lippe, who did all the bioinformatics and computational biology that I presented today. Alessandro Sette, who is very important for all the project, the part related to the binding to the MICHI class 1, and Ellis Seitz, who led the Listeria monocyte genes project, and also CR UK and the other funding that reported that fund my research. And I would like to finish just advertising the fact that I am recruiting at the moment one postdoc for a Cancer Research UK Program Foundation Award. The project just started. The call will close in around 2 weeks. If you are interested in what I'm doing, please don't hesitate to contact me. And if you are more interested in dry lab than wet lab, a colleague of mine, Juliane Lippe, at the Max Delbrück's at the Max Planck Institute in Göttingen, has 3 postdoc positions funded by her ERC starting grant. With this, thank you everybody, and I stop sharing.
That's great. Thank you very much, Michele. Thank you to both speakers. So Lindy, if you could just press your webcam so that we can again see you. I will go through some of the questions, but please carry on asking questions. So the first few questions are to Lindy. The first question is, if the epitope repertoire, i.e. CD4s and MHC expression, exist in cancer patients, why is this not stimulating a tumor-restricting immune response naturally? How would vaccination change this?
That's a really good question. So we do have some evidence to suggest that some of these responses are indeed memory responses. And the argument would be that it's to do with the tumor immunosuppressive environment, or indeed exhaustion of those cells that can no longer be effective. But what we've shown is if we then go in with the vaccine basically generates a large number of very active CD4 T cells, and they can overcome that hurdle.
Okay. I'll ask you a couple of questions, Lindy, while people are asking questions from Michele. Are the citrulline-related peptides from different humans sort of similar? I mean, in other words, how do you hope to have something that'll work on a single vaccine against many different cancers? I think is the basic question. Yeah,
again, very good question. It seems to be that there are dominant epitopes or dominant areas that are citrullinated, and clearly that then depends upon their ability to be HLA bound or restricted. So far, the momentum epitopes that we've looked at and the inalase epitopes seem to be expressed by all the mouse tumours. So to a certain extent, they do seem to be fairly ubiquitously expressed. A little bit more difficult to tell with the human cancers because clearly they don't express MHC2 unless you induce it, and then you have to induce it under stressed conditions. So at this point, we'll see with the clinical trial how ubiquitously they are expressed.
A question for Michele, maybe. What can you estimate what proportion of peptides that are being presented by a cancer cell might be constituted by these spliced? I mean, is it likely to be a large proportion or just a very small proportion?
So the frequency of the spliced peptide in immunopeptidome is a very controversial topic. There's been many meta-analyses since our publication in 2016. The results, they are strongly dependent on which method you use, and then some groups say that is below 0.1%, and other that is reached 30%. I think that all studies so far, including ours, have made the mistake that they didn't really benchmark the performances of the methods. So we always assumed that what we were seeing was everything correct and was the only things that could be seen. That is, I think, the incorrect approach. So at the moment we are developing a method that, on the contrary, is being strictly benchmarked, and our estimation of the cis splice peptide that derived from the combination of fragments that are separated of a maximum of 25 amino acids. So this is one part of all the splice peptides are around 10%.
Thanks. Lindy, again, do you see a question for you? Do you see any evidence of epitope spreading, or do you think this is unlikely given the lack of a CD8 response?
Again, very good question. We have not seen any evidence of epitope spreading. So if we immunize with one citrullinated epitope, we don't see cross-reactivity to any of the other citrullinated epitopes. So we've probably got about 30 epitopes we've characterized now. Or indeed, if we give them tumors and then immunize and look for regression, their immune responses in the spleen are still very antigen specific, i.e. specific to the immunizing vaccine. But clearly, these are in the HLA transgenic mice. I don't really understand what will happen in humans.
Right. There's a question about macrophages. Do you have any evidence at all that there's a role for macrophages in the response, in the protective response?
So the only work we've really done is on the sort of the myeloid-derived suppressor cells or non-suppressor cells, so more myeloid really than macrophages per se. And as I say, we do seem to see loss of myeloid-derived suppressor cells. We don't see a subsequent gain in basically more activated or M1 type macrophages. But their studies are quite difficult to do in these regressing tumours where we have limited amount of material. So potentially, but still to be proven.
Right. And another sort of cell question. I mean, is there actual evidence that the CD4 cells that you're seeing are cytotoxic CD4 cells? For example, do they have granzymes and that sort of thing?
So they definitely have granzyme. So that's what we were showing you in both the mouse and the human. They express granzyme. We have not been able to show direct killing. So I don't know whether that's a failure of the assay or whether they're using granzyme in a different way.
Okay. A question for Michele. How transient or stable are spliced peptides across a single tumor? Might some cancer cells present spliced versions and some not within the same cancer?
Yeah, this is a very interesting question. So the impact of the metabolism on the generation of the splice peptide still has to be investigated. So it could be that there are difference from different, so in intratumoral differences, as well as if there are clear difference between a normal cell and cancer cell. But this is one of the things that we want to investigate in the future.
Okay. Sorry, I'm just scrolling through there. A lot of questions. And a question for Lindy. You've picked triple negative breast cancer for the clinical study. Is this the best breast cancer subtype in terms of MHC2 expression and expected response?
It's probably picked more on the fact that it has high levels of EMT. So in essence, during epithelial to mesenchyme transition, that vimentin is degraded. So it's expressed and then degraded. And we think that makes it a very good target for autophagy and presentation. I was surprised at the level of MHC class 2 these class tumors had, these triple negatives had, so that's a bonus, but it was really picked on the fact it's a really good EMT tumor for vimentin.
Okay. And for Michele, has anyone identified any other spliced peptides for other KRAS G12 mutations, such as G12C or G12D? And if so, can they be efficiently processed and presented?
Yeah, this is one of the topics of my CRUK project.
So it's not yet answered, in other words.
Not yet.
Okay. Lindy, is there a risk of triggering RA in response to the citrulinated peptides? And a sort of related question, maybe I'll put them together, is what's the actual, is there a biological function for this citrullation which could be then disturbed, obviously, if you use citrullinated peptides as an antigen?
Yeah, both very good questions. So there is always a risk potentially that we could induce RA, although RA is mainly associated with antibody responses to basically deposited proteins, citrullinated deposited proteins within the joints. So you see anti-citrullinated antibody responses, and it's those immune complexes of antibody and antigen that basically drive the rheumatoid arthritis. Because we're using relatively small peptides, we don't see an antibody response. So we exclusively see a T cell response. So we think maybe that's why we're not inducing rheumatoid arthritis. That said, in the clinical trial, clearly we will be very careful about putting anybody with any sort of autoimmune in there initially. And we will monitor very closely for rheumatoid arthritis. And in answer to your second question, I do believe this is a stress response. So I think it's a response to a combination of stress and inflammation. So these responses could be well triggered during some sort of infection procedure as well, where you're stressing cells and you're also upregulating MHC class 2. There could be other situations where you get stress but not inflammation, and that would not be an issue. But as long as you get stress and inflammation, potentially they could be recognized, and that's their normal function. So far, we've seen no toxicity in the animals. So we've done large numbers of studies, even these animals that regress the tumor and then we re-stimulate and they regress again, don't show any sort of side effects. But again, I have to admit, this is in animal models, a relevant animal model, but whether we see the same in humans, another question.
Great. We're almost out of time. Maybe a quick couple of questions for Michele. What's the ideal motif length of splice peptides that might have better binding affinity and therefore may have potential to be used as a vaccine adjuvant or a vaccine?
Well, regarding the sequence preference for the splice peptide and how this is combined with the MHC class 1 binding affinity, we are working on that. So there are like evidences, but still we don't have the full picture.
Okay, great. And are there any examples from infectious diseases? One last question, maybe, where there's evidence for a splice peptide as an antigen?
Yeah, so we had the study on Listeria monocytogenes, and on virus there is Seth Borrow at Oxford who published a study on HIV on human patients last year.
Right. Great. Well, I haven't been time to read all the questions, almost all, but we're sort of running out of time. So let me just finish off by thanking both speakers. Really fascinating talks. Thank you very much. Thank you for all the attendees. There were nearly 400 people attending, so that's a great number, and some excellent questions. So thank you very much. Hope everyone enjoyed it. There will be a recorded version of the live webinar available on the Regional and Affinity Group section of the BSI website, so you can see that there. And there'll also be a link to this as soon as it's available. Just a foretaste, there'll be another webinar on the 4th of March entitled "What Do Cancer B Cells See?" So sort of the other side of the picture, also hosted by the Tumour Immunology Group, and you can sign up for this on the BSI website. One last thing, all attendees will receive a feedback survey shortly after the end of the webinar. Please do take just a minute or two just to fill it in so that we can hear your views and improve the webinar. So thank you very much indeed again, and hope to see you at some future webinar. Bye.
Thank you very much. Bye-bye.
Thank you.